number 18. And the idea here is to apply some of the things that we learned already about DNA and some of the things we've learned already about evolution to try to come up with some ideas about how we go from one species to a new species and how speciation could happen, some of the mechanisms of speciation. The idea in the last couple of podcasts where we're talking about how we get to a cell, it's jumping a whole lot from a cell to, say, something like a fish or something like that. But the idea is that once we had organisms on the planet, however that happened, scientists have some ideas about how that happened, as I've talked about in the last couple of podcasts. Then how is it that speciation happens? You go from one species to another. And the short answer is we don't know. We do know, but nobody agrees on one common answer about what defines a new species. So we have identified mechanisms by which speciation could happen, but not how much, and that is involved with genetic diversity, basically, changes in gene sequence. But how much gene sequence means a new species, that is something that is usually defined by biologists who specialize on studying how speciation happens, taxonomists. And those biologists typically specialize on a certain group, this is an ancient plant, a fern, and within that group, those scientists, when they publish a paper about how many species there are in a particular group of lower plants, would make an argument to the rest of the scientific community saying, I saw this much genetic diversity and I'm calling that a new species. And that's pretty much how it happens. But because we don't totally agree on it, and the rules by which we collect genetic information and how we understand mutations happen, those rules change over time, and the technology changes over time, so that sometimes we totally revise what is a species or what is not a species, and how species are related to each other. If you looked at a textbook from the 1980s and a textbook from today for biology, it would have pretty big differences in how organisms are grouped and what's a species and what's not a species. So the bottom line of this podcast is that even though we know mechanisms, and we'll talk about those mechanisms by which genetic differences are generated, and those genetic differences can lead to speciation, how much genetic difference equals a new species is something that scientists argue a lot about and agree on within a group. So people who study fishes, for example, or people who study plants might, for example. So this picture this is a fossil fern. It looks very much like a modern day fern from what we can see of the structure on that fossil. But we know that millions of years have passed between this and this. Are these the same species or not? And how would you decide? Okay. In modern definitions of species, when it's possible, they collect genetic information from the things that they're trying to classify as species or not species, and then they subject that genetic information to analysis. In the case of fossils, sometimes it's possible, but not always possible, to get genetic information out of fossils, and so they use other characteristics besides genetic information, like the pattern of the leaves, or where they have the reproductive structures called sori, that sort of thing. And if a biologist can show that gene flow has been restricted, that within this group, they're only mating with each other and they're not sharing genes with another group, that would be usually enough to define it as a new species, depending on how long they've been separated. Sorry, you're just hearing my cat there. Okay. Let's go back to Mendel's peas. In those true breeding characteristics that we noticed, or that we talked about, such as flower color, pea shape, there was another one, and that was the height of the plant. And he noticed that either you got tall plants or short plants. Tall plants as high as 6 feet tall, and short plants only about a foot tall. And he knew this is a true breeding characteristic, and didn't know the genetic basis for it. Mendel didn't know anything about genes. The word gene didn't exist. So after all these famous studies by Mendel, other scientists have found the genetic reason for this height difference. And this is part of the question that I introduced on the first slide, is this difference in shape enough to say that these are different species or not? In the very first couple podcasts when we talked about dogs, certainly the shape of dogs can be really different from like a Chihuahua to a Great Dane, but they're all considered one species. So it's not always just on what they look like. Okay. This gene that's involved in the height of peas is called LE. And if you go on GenBank, and there's a exercise in your book with the links that you can go on GenBank, and there's a GenBank is an online depository of all kinds of gene sequences, and you can click on those gene sequences, and then you can compare the two genes, the tall version of the gene and the short version of the gene. The gene codes for a hormone that is involved in cell division, and cells that have plants that have a lot of this hormone divide more rapidly, and they're taller, and less of this hormone less rapidly. Okay, you can, in this online tool called BLAST, which stands for Basic Local Alignment Search Tool, you can compare sequences one to another. You can say, are these sequences the same or not? And it'll line up two sequences, and you can see every place where there's a line, the sequences are identical. And first glance, they look completely identical. Okay? But if you compare more of the two sequences, the short version and the tall version, there's one spot, just one spot out of 720 bases that is different. So these are the one-letter codes for the nucleotides, and one version in the tall version of the plant, there's a G at this position. In the short version, there's an A at that position, and that switch causes a different codon to be coded for. It's GCC in the tall plant, ACC in the short plant. GCC codes for alanine, ACC codes for threonine. So it makes a different protein, and a different version of that protein causes the shortness in the plants. So how can you get those sorts of mutations? So I casually mentioned in another podcast that you can, be exposed to the sun, cause a mutation or that sort of thing. More mutations come from the action of DNA polymerase. That's drawn as this big blob here. DNA polymerase is the enzyme that copies DNA. When DNA goes through semi-conservative replication, you get the two strands. This would be the bottom strand, and then there's a top strand here that they're not showing you. They unzip, and then one strand acts like a template to make a new strand. There's a short RNA primer when this is made in the cell. When it's done in a test tube like you're going to do, or maybe you'll talk about in lab, because it's online this year, they use DNA as a primer just because DNA is more stable than RNA. Then DNA polymerase uses this short piece of primer. Primer is just a short piece of nucleic acid, RNA, or DNA. Most of the time in the lab we talk about DNA for primers. But when it's the DNA is duplicated during the S phase in the cell, RNA is a primer. Okay, primer is a short spot, and that's the only place on this unzipped strand of DNA where the two strands are separated. That's the only place where it's double-stranded, and that's how DNA polymerase knows where to go. And DNA polymerase will zip along and read the strand on the bottom and say, okay, if there's an A here, I'm going to put a T here. If there's a C here, I'm going to put a G here, and so on. And it'll copy pretty fast, thousands of base pairs per minute when it copies that DNA. But occasionally it'll make a mistake. It'll make a mistake, say 1 out of every 5,000 base pairs or 1 out of every 10,000 base pairs, and that sounds like that's pretty good. That's not many mistakes. But if you consider that the human genome is 3.1 billion base pairs, if you make a mistake every 10,000 base pairs, that's actually a lot of mistakes every time you copy your DNA. So those mistakes where like there's a G here and instead of putting a C here, you put an A instead, that can cause this kind of mutation, and it can have an effect on the organism, like making the pea plant short. Okay, it can also have no effect at all. It all depends on where the mistake happens. Okay, so DNA polymerase makes mistakes, and that's where a lot of mutations come from. If I'm trying to compare those two sequences, I can do this sort of thing, an alignment, where I'm aligning those two sequences, and I see just this one spot where they're different. Okay, there's no bar right there. I can also compare sequences in what's called a dot plot. There's a big section of your book shows you how to draw dot plots, but we'll talk about them here. For small changes, like that one base pair change, dot plot's not that great. But for bigger changes, you can get a better idea. So if you have two sequences you're comparing, okay, the 5' end of one of the sequence starts here and it goes toward the 3' end here, and the 5' end of the second sequence is here and it goes toward the 3' end there. And every place where they are the same sequence, you put a dot, okay, in this grid. So number 1 is A, number 1 is A, put a square there. Number 2 is C, number 2 is C, I put a square there. Okay. Number 1 is A, or number, 1, 2, 3, 4, 5, 6, 7, 8, 9 is A, and number 1 is A. So I will put a square there. Okay. So when you fill out the dot plot looking for all the matches between the two sequences, it's not so useful. But if you say, okay, now instead of looking at every base, one base at a time, I'm going to look at, say, 5 in a row. I'm going to look at these 5 bases, A, C, G, T, G. And is that the same as these 5 bases, A, C, G, T, G? Okay, A, C, G, T, G. And I get a yes. And so I draw a dot right down the middle. And then I move that window of 5 to the next 5. 1, 2, 3, 4, 5. And I say, are the two the same? And if the answer is yes, I put a dot there. Okay? So if the two sequences are pretty similar, almost exactly the same, you get a line down the middle, and that's a way of just looking at it and saying, okay, these two sequences are similar versus looking at that big chart where all the sequences are lined up. Okay, this is called sliding window because I have a window of 5 that I'm sliding around along, one nucleotide along, but I'm looking at 5 at a time. These 5, and then these 5, and then these 5, so on. Okay, I can change my window. I can change it to 20 and say look at 20 at a time. And there are shorter stretches of this sequence where there are 20 nucleotides that are the same in a row on both of them for that amount of time. And if I make the window smaller, I get more of the sequences the same for 13 in a row. Okay? If I'm looking at it and I get a break like this, okay, that is an indication that there has been sequence deleted or inserted in this spot here in the middle someplace. So they start off being the same, then things go screwy, and then they end up being the same again. Okay, and that I can tell just by looking at this kind of plot. If I look at something more realistic where I'm looking at a lot more sequence, okay, from 1 to 4.6 million base pairs, okay, and from 1 to 5.5 million base pairs, they don't have to be the exact same size, but for a stretch of this sequence, it looks like they're really, really similar, and then there's a break, so there could be an insertion or deletion, and then they're similar again. And these are 2 different chromosomes from E. coli. So the overall trend is that these 2 chromosomes are really similar. The sequence is really similar in these 2 sequences. But there is a shift in there, and it could be because this organism here lost DNA, or it could be that this organism here it could be that this organism here lost DNA. It could be that this organism here added some DNA sometime in its history, causing that break. And you can't really tell by looking, but you do know that there is a break in there. When you have a reverse slope, something like this, it indicates that the sequence is in reverse orientation. So it was cut out, flipped around, and stuck back into the sequence somehow. And to indicate that on the diagram, you just color it a different color and say, okay, there's been an inversion in that part of the genome. And then if you have those parallel lines, which we saw on the last simpler diagram, it indicates that parts of the genome have been duplicated. So that's one of the mistakes that DNA polymerase can make, is it can skip like an old-fashioned record, or I guess for you guys like an old-fashioned CD. While it's reading the information on one of the strands, it can skip and recopy the same section of DNA more than once. And that creates duplicated DNA segments. Sometimes this happens with a whole chromosome, and a whole chromosome gets duplicated. But that's a pretty common occurrence that DNA polymerase causes. So, and there's a couple duplicated genes that have been inverted. So is these differences, this here and the break and the parallel lines and so on, the duplications and the inversions and the insertion of DNA, is that enough to call this 2 different species when we're comparing this species and this species? You'd have to talk to a microbiologist who studies these to see if that much genetic difference was enough to call them two separate species. Okay, again, it is defined within the field of people who are studying how these organisms are related to each other, and they have to make a case to the other scientists that this much genetic difference is a new species. Okay, so again, if we're looking at these plots, you put the 2 genomes next to each other with the origin, the 5' nucleotide in the same spot. one going 5' to 3' this way, one going 5' to 3' this way. And then you plot all the sequences and use a sliding bar to see where the matches are. When you see this sort of a pattern against the main slope, that indicates there's inverted DNA, that it was cut out and switched around. The 5' and 3' ends switched around and then reinserted. This could be an inserted piece of DNA or a deleted piece of DNA, and this would be a duplicated piece of DNA in the genome. Another way you can look at genomes is with color maps. So this is the E. coli genome. Remember, it's one chromosome, and it's in a shape of a circle. It's double-stranded, so they're showing you one strand here and another strand here. And they're color coding it for, this is lots and lots of sequence, and within one of these little tiny bars is a whole gene, and in that gene they're measuring the percent of the gene that is G or C. So there's A, C, G, and T in the nucleotides. If I'm say the gene goes from 1 to 100, and say how many of those 100 base pairs are a G or a C, and calculate that percentage. And then you give it a color according to the percentage. So it's a 50-50 GC and AT, it's going to be yellow. If it's higher percentage GC, it's going to be red. Lower percentage GC, it's going to be green. Okay. Some genes in here you can see are outliers, they're red. Some are green. Overall, if you're going to say what is the color of that circle, most of you would say yellow, some version of yellow. So overall, this genome is about 50% GC. And if you see something that is red, like this, or green, like that, that seems to be an outlier. And then black is tRNA, so that doesn't count. It's just how they color-coded it. They didn't. Certainly, tRNAs have A's, C's, G's, and T's too. They just color-coded it differently. All right. So why look at it this way? There can be something called horizontal gene transfer, where one organism contributes their genes to another organism's, and they become part of their genome. And one way to tell if this gene came from another species is by this sort of color mapping. If most of the genome is yellow, and then you have a couple that are really different color, maybe those came from another species of bacteria. And bacteria are pretty famous for collecting genes from their environment, collecting genes from from other organisms. This is how they get antibiotic resistance, or that's how they can transfer antibiotic resistance. But even humans have horizontal gene transfer. Some of the viruses that have infected us in our evolutionary past are now part of our genome. And if the whole genome is just A's, C's, G's, and T's, if you're looking at sequence, how can you say, oh, this sequence is different than the rest of it? It all looks the same. Well, this is one way you can tell, in that if most of your genome is 50% GC, and then you run into a stretch that is way higher or way lower than that, it's likely that that gene came from someplace else. In the mouse genome, 1% of its total genome is originally from a virus, is estimated. So that's one way you can look at genomes. Another thing you can look at is just how much DNA is there in the cell. So we talked about diploid cells when we talked about mitosis and meiosis, where you have two copies of everything. One came from mom, one came from dad. Okay? After replication, But before cell division, those chromatids go from 1 chromatid per chromosome to 2 chromatids per chromosome, so you get twice as much DNA before that is separated again during anaphase. But then there are some cells that have even more than that, lots more DNA than that. So where could that come from? Okay, if they look at the number of chromosomes in this animal, and this happens to be a mouse, it's supposed to have 40 chromosomes. And in wild type is another name for quote-unquote normal. Most of the cells do have 40 chromosomes, but some have way more than that. So 40 chromosomes in wild type cells. Okay, this is in mice, not humans. So humans would be 46, but 40 in wild type cells. But in this one cancer cell line, there is some as many as 100 chromosomes in a single cell. So one of the things about cancer is uncontrolled cell growth, and cancer cells will duplicate their DNA and then never divide. And so you get abnormal numbers of chromosomes in there. So if you have, remember we talked about in anaphase that you have 2 chromatids per chromosome, and that splits into 2 chromosomes, each with 1 chromatid. If that doesn't happen, and you don't do that split, or you don't divide the cell by cytokinesis when you're done, then you get twice as many chromosomes per cell. So if you're not going through the normal cell process, like in what sometimes happens in cancer cells, you can get extra chromosomes. And sometimes this can happen in organisms' history, and the whole genome will duplicate. This is a little fish called fugu. It's a puffer fish, and it was one of the first vertebrates, things with a backbone, that had its genome sequenced, because its genome is fairly small compared to other genomes. And so because it was small, it was easier to do. And even within the fugu genome, there's all kinds of gene duplications that have happened during its history. And so there are more than one version of a gene in a genome, and those are called paralogs. Okay. To talk about paralogs, let's say we have 2 different species. We have rectangles here by this teal rectangle, and we have circles here by this yellow circle. Those are 2 different species. And let's say that in this species you have, well, so let me back up for a second. So paralog is a duplication within a species. So you have more than one version of the gene within a species. And orthologs are versions of a gene, the same gene in different species. Okay? So if I have, let's say we're talking about the hemoglobin, one of the hemoglobin genes. Hemoglobin is the thing that is a protein that carries blood, or carries oxygen in your blood, and you have more than one version of hemoglobin, and they have arisen by duplication within humans. But we're not the only things with hemoglobin. Dogs have hemoglobin. So if I was saying my adult hemoglobin versus my juvenile hemoglobin, those would be paralogs. So they're duplications within a species, within humans. But if I was comparing my adult hemoglobin to a dog adult hemoglobin, those would be orthologs. Those would be the same version of the same gene in different species. Okay, let's illustrate what that looks like. Let's say we have chromosomes 1, 2, and 3. 1 is pink, 2 is white, 3 is black. And this is a diploid species, so it has 2 copies of everything. One from mom, one from dad. One from mom, one from dad, one from mom, one from dad. In this other species, they're tetraploid. They have 4 copies of everything. So 2 from mom, 2 from dad, 2 from mom, 2 from dad, 2 from mom, 2 from dad. Let's look at this gene A. This species has 2 versions of A, called A1 and A2. Those are called paralogs because they're within a species and they're 2 different versions of A. And then also has 2 different versions of B. But it only has 1 version of X, okay? And this tetraploid species over here also has a version of X. So that would be an ortholog, the version of X in one species versus the version of X in another species. These here, okay, between the arrows, this one here versus this one here, this one here, this one here, this one there, this one here, those would be called alleles. Remember we talked about those when we talked about meiosis, in that they are the version of the gene. And if this diploid species was going through meiosis, it would take either this allele or that allele going to a sperm or an egg. Okay? All right. So, back up for a second. This idea of gene duplication and genome duplication, okay, where the polymerase, as it's duplicating the DNA during the S phase, skips and copies the same thing over again. This is an important feature of evolution. It's a common occurrence. Almost all species have paralogs and orthologs. Okay. What this allows is that if gene A is vital, you got to have gene A or you die. Now you have 2 copies of gene A, and if one of them acquires a mutation that would normally kill you, then the other version can take over. Another thing is that you always have one version of A to keep things going, and then the other one can accumulate mutations. Most of them will be bad and will cause the gene to no longer produce a functional protein. But every once in a while you might get lucky, and it may make a version of the gene that gives that organism an advantage, a selective advantage. And then natural selection can select for that new mutation, that new allele, because it gives that organism an advantage. And then that organism can have greater reproductive success. So if we go way back to the beginning, and we talk about our short peas and our tall peas, this little mutation that showed up, causing peas to be short, maybe they're now in an environment where there's something feeding on these pea plants, and they always go for the tall plants because they don't have to bend over as far to eat those, and the short plants get spared. So the tall plants get wiped out just because they're closer to something that's eating them, their mouth, and the tall plant and the short plants have an advantage in that situation, in a natural selection situation, so that they get selected for. They leave more pea progeny into the next generation, and then the percent of alleles that carry the short allele in the population goes up over time. That is an example of natural selection. Okay, and how these gene duplications can then be acted on by natural selection. All right, I just want to leave you with this at the end. We started with how do you define a new species, and I told you right away we're not going to give you the answer. So there is no one answer. There are mechanisms by which we know that mutations happen, okay, by mistakes being made by DNA polymerase, as an example. Those can lead to new versions of genes being made. If the gene is duplicated, and both versions are in the same organism. Those are called paralogs. If we're talking about the version in one species versus the same thing in another species, that's called an ortholog. That can be acted on by natural selection, and natural selection may cause enough of a genetic difference so that over time you have two different species. How we decide in modern times what is a species and what is not a species is by taxonomists who study the organism. Almost always they're using genetic information if that's available, unless they're studying dinosaurs. And then they use that genetic information to say there is this much distance genetically between these two organisms, and I'm going to call them a separate species for these reasons. Okay, but science changes their minds all the time about what is or is not a species within a group, and sometimes major divisions that have stood for a long time collapse. So, like when I took this class, there was only 3 kingdoms. Now there's 5. So the more we know, the more this will change for you as you study biology.